Okay, welcome back to this next video in which we are discussing the effects of sympathetic stimulation on the heart, and we're specifically concentrating on the chronotropic effects, i.e. The increase in the heart rate that results from the sympathetic stimulation of the sinoatrial node. Okay, right, so I gave you these concentration gradients and gave you this conundrum of whether sodium is going to move in, or, well, what the net movement of charge is going to be through these HCN channels. Okay, so sodium, the concentration gradient is favouring the movement of sodium inwards. Okay, so there's a 12-fold concentration gradient. That means that sodium is 12 times more likely to hit from the extracellular side and go in than it is for a sodium ion to hit from the intracellular side and go out. So there's a 12-fold gradient inwards, so you're 12 times more likely to get the ion moving in than moving out, so you're going to get a net movement of sodium in, just from the concentration gradient. In addition, you've got a lower electrical potential within the intracellular compartment, lower by 70 millivolts. Sodium is a positively charged ion. It wants to go where the electrical potential is lower, which is the intracellular compartment. So you're going to favour the movement of sodium in because it's being pulled in now by the electrical gradient as well. So that's going to increase the net movement of sodium in even more. Okay, so we've got a net movement of sodium in. Okay, now potassium. You've got a concentration gradient of around 40-fold, favouring the movement of potassium out of the cell. Okay, so we would expect to get potassium moving out, or would we? The electrical gradient now. The electrical gradient is negative 70 millivolts. The electrical potential of the intracellular compartment is lower than the electrical potential of the extracellular compartment by a whole 70 millivolts. Potassium, too, is a positively charged ion, so it wants to remain where the electrical potential is lower, which is the intracellular compartment in this case. So the electrical gradient is favouring the movement of potassium in, the concentration gradient is favouring the movement of potassium out. Which wins? Well, at negative 70 millivolts, it is just the concentration gradient that wins. Indeed, if you wanted to actually match the concentration gradient, the electrical gradient would actually need to be bigger than that. It would need to be negative 85 or there around. However, the movement of potassium out is very small compared to the movement of sodium in. So the main important thing about these HCN channels is that when they open, you're going to get movement of sodium ions into the cell. Now, what is that movement of sodium ions into the cell going to do? Well, sodium ions have a positive charge. So you have effectively got a current, a positive current coming into the cell. You are moving positive charge into the cell. And in fact, this current has a very famous name. It's known as the IF current, or the funny current. Okay? So, the funny current is just this movement of sodium in through the HCN4 channels. The HCN channels, of which the main example in the sinoatrial node cells is the HCN4 homotetramer. Now, when this happens, you're going to bring in this positive charge, and you're removing it from the extracellular compartment. So, when you bring in positive charge into the intracellular compartment, that's going to raise the electrical potential of the intracellular compartment. So, the electrical potential of the intracellular compartment is going to go up, and it's going to reduce the electrical potential of the extracellular compartment because you're removing the positive charge from the extracellular compartment. So, initially we were at negative 70 millivolts, so that meant that the intracellular electrical potential was lower than the electrical potential extracellularly by a whole 70 millivolts. But now we're raising this and reducing this. So the amount by which the intracellular compartment's electrical potential is lower than the extracellular compartment's electrical potential is going to get smaller. So what you're going to get, basically, is a depolarization, a steady depolarization as the funny current comes in. Okay, so what happens is you go up to around negative 60 millivolts. Now, when the electrical potential difference across the membrane gets to negative 60 millivolts, that's the threshold potential for the activation of another channel. Okay, so let's draw this next channel that's going to come into play here. This is what's known as a T-type voltage-gated calcium channel, which I will show in blue here. So let's have a little discussion of voltage-gated calcium channels further down here. Okay, so we want to know what is meant by a T-type voltage-gated calcium channel. And voltage-gated calcium channel is often abbreviated to VGCC for short, for voltage-gated calcium channel. Okay, right, so voltage-gated calcium channels. Voltage-gated calcium channels are not just one protein. They are a huge number of proteins all stuck together. However, there is one protein in this mess that is the most important protein. So if we have the membrane here, so this is the phospholipid bilayer. So there is one protein which actually makes the pore-forming subunit. So it's the portion that actually has the pore which allows calcium to move through it. And this subunit of the voltage-gated calcium channel is known as the alpha-1 subunit. Okay, and if you just make an alpha-1 subunit with no other proteins, no other riffraff to join on, it's a perfectly functional voltage-gated calcium channel. Okay, so this is the most important subunit for a voltage-gated calcium channel. So, in the human genome, there is not just one gene for an alpha-1 subunit. There are in fact 10 genes for alpha-1 subunits. So, we have conveniently grouped them into families. So there is the CAV1 family, standing for voltage-gated calcium channel family 1 of genes for alpha-1 subunits. Then there is the CAV2 family, and finally the CAV3 family. And we've grouped the 10 genes into these different families. Now, in the third family, there are 3 different genes which all code for alpha-1 subunits. Now, all of these 10 genes that we've put into these families, they will all have slightly different sequences of organic bases, and they will code for slightly different proteins. However, the protein performs a similar role, so similar that we call them the same protein, even though they are actually different proteins if you look at the primary amino acid sequences. But they perform the same role, basically. So, in the CAV3 family, we have 3 genes. The CAV3.1 gene, the CAV3.2 gene, and the CAV3.3 gene. Okay, so these are the 3 genes of the CAV3 family, and they all code for alpha-1 subunits. Now, if you use one of these 3 genes in the CAV3 family to make your alpha-1 subunit of your voltage-gated calcium channel, then the voltage-gated calcium channel is henceforth known as a T-type voltage-gated calcium channel. So that's what it means to be a T-type voltage-gated calcium channel. It means that your alpha-1 subunit is one of these 3 genes, well, it was made by one of these 3 genes. Now, what does this matter? Why do these 3 genes actually encode for a protein that does anything different from these ones? Well, yes, is the answer. The T-type, these are called the T-type because T for transient, okay? And the voltage-gated calcium channels, which are produced from alpha-1 subunits from these 3 genes, they open very, very rapidly and they close very, very rapidly. So they're open for a very transient period of time. Now, that property is very different from some of the other genes that are in the CAV1 family and the CAV2 family. So it does matter, basically, that we're using this different alpha-1 subunit. Okay, right. So, we'll colour in the alpha-1 subunit red. Now, as I said, voltage-gated calcium channels don't just consist of the alpha-1 subunit. They also have a bunch of auxiliary subunits, which I'll talk about now. So, these subunits aren't as important as the alpha-1 subunit, but they do moderate the function of the alpha-1 subunit and change its properties slightly. So we have the gamma subunit in violet pink here. Okay, so this is the gamma subunit sitting next to the alpha-1 subunit. Here we have the beta subunit sticking off the intracellular aspect of the alpha-1 subunit, so this is the beta subunit, and over here, in blue, is the alpha-2 delta subunit, and that's really two subunits that have been stuck together. So the alpha-2 bit is this box at the top, the delta bit is the membrane straddling bit. Now, the reason it's referred to as one subunit rather than just as two subunits stuck together is because actually the alpha-2 subunit and the delta subunit are encoded by the same gene. So the gene for the alpha-2 delta subunit makes the protein, which is then cut in two, and those two proteins take on their form, i.e. the alpha-2 and the delta subunit, and then those two subunits join back together, but in a different way to the way that they were originally joined together as a polypeptide. Okay, it's actually by disulfide bonds that they join together. Right, so that's a T-type voltage-gated calcium channel. Now, in the next video, what we'll see is what its role in the action potential of the sinoatrial node cell is.